now we will introduce DNA replication. As a reminder, this occurs during interphase, and it's in that S phase is when DNA replication will happen. Some background information on DNA. We know that it is made up of our monomer of nucleotides. This is our nucleotide. We have a phosphate molecule, a sugar, and a nitrogen base. And it could be one of 4 different options: adenine, thymine, cytosine, or guanine. So look at a DNA model right here. It's that twisted double-stranded helix, and we have our phosphate, our sugar, and then our nucleotides that connect. So we can say this is cytosine, and it will bind with guanine. So how does it do that? It uses those hydrogen bonds in between those 2 bases to hold together the 2 strands. As a reminder, we have a couple of enzymes that are involved in this process. We have helicase, primase, DNA polymerase, and ligase. If we want to look at an example strand, so say we take this portion and this is what it looks like. Adenine will always pair with thymine, and it has 2 hydrogen bonds holding them together. Cytosine will pair with guanine with 3 hydrogen bonds. And again, thymine with adenine and adenine with thymine. So if this is our double helix, for example, our first one to come in would be helicase. That enzyme will unravel it and then pull it apart. So by the time helicase is done, you have 2 separate strands, and they're complementary but not identical. The next thing you'll do is you'll take these 2 strands, and these are our original template strands, and they're going to bind with new complementary strands. So primase will come in and prepare that strand, and then DNA polymerase will come and add bases to the opposite side. So then adenine pairs with thymine with 2 hydrogen bonds, cytosine with guanine with 3 hydrogen bonds. It'll do the same for our opposite template strand. So they're all paired up, and then ligase comes in at the end. We'll start it off, and it will clip those 2 strands together. So then by the end of it, we have one side that's the original and one side that's our new strand. And it does the same for the opposite one. So by the time you get to the end, you have 2 double helixes, and these 2 double helixes will be identical. And this is called semi-conservative because there's 1 old strand and 1 new strand.